In this video, we're actually going to look at the process of translation, which is where the tRNA, mRNA, and enzyme component of the ribosome, the rRNA, actually work together in order to assemble the primary structure of a protein. So if we start at the beginning, what we call translation initiation, what we want to do here is assemble all of these components into the fully functional structure that is actually going to make the protein. And the start of it is where our transcript mRNA binds to the bottom part of the ribosome. And we can see in this diagram that the ribosome is not actually a single structure. It is actually made of two different subunits. So the subunit at the bottom is what we call the small subunit of the ribosome for obvious reasons. And on the small subunit, we can see that there's actually an active site here that specifically recognizes mRNA and allows it to bind. And when this happens, the small subunit is going to move along the mRNA from the 5' end towards the 3' end, and it's going to look for the three bases AUG. Now, as we looked at when we talked about codons, AUG is significant because this is the start codon, and therefore the start signal for the ribosome to actually start working, and it's also the signal for the ribosome to input a specific amino acid, which happens to be methionine. Now, the reason why the ribosome is able to recognize this site here, the start codon AUG, as to input a methionine is because the tRNA molecule that is carrying methionine is specifically able to recognize this codon here, because at the bottom of the tRNA molecule, it has a structure called an anticodon that specifically matches the three bases of the codon. So from the name anticodon, anti obviously means opposite, meaning these three bases here, UAC, are the exact three bases that would be opposite to and therefore match the three bases of the codon AUG. And this is how a ribosome knows which tRNA inserts which amino acid at which point on the protein because of codon-anticodon base pairing. Now, once the first tRNA molecule is actually in place, this allows the large subunit, also known as the subunit that is on the top of the ribosome, to now attach to the protein synthesis complex here, because the ribosome here is able to actually grab hold of the first tRNA molecule as it has entered one of the three active sites that exist in the ribosome. So on that note, what are the role of the three active sites, and why does a ribosome actually need three? Well, on that note, we can actually start talking about the process of protein synthesis, also known as polypeptide elongation, taking one amino acid and elongating it until we have a polypeptide. So on the subject of each of the active sites has a distinct name, the first being the A site, the second being the P site, and the third being the E site, where some of the names are quite intuitive based on what their role is, and others we need to discuss. So the A site is technically where translation begins. This is what we call the rather awkwardly named amino-acyl tRNA site. Now, this is the location where, as long as a tRNA molecule is bonded to a specific amino acid, that tRNA is able to enter the A site and be ready to undergo a chemical reaction to elongate the polypeptide. There is only one exception to this. The only exception to all tRNA entering the A site is the tRNA that actually matches the start codon, because as we can see on the previous page, that tRNA molecule did not enter the A site, but actually the large subunit dropped right on top of it using the second site that we call the P site, which is what we call the peptidyl or peptidal transferase site. And the name for that kind of tells us a little bit about what this site does. we can see the root of this name is transfer, which means that something is going to be passed from one location to another, and we can see the suffix ase actually indicates that it's referring to the catalytic part of the enzyme, the rRNA part, also known as the ribosome. And this is where the catalytic part of the ribosome actually grabs hold of the growing polypeptide chain that is produced and actually transfers this from one tRNA molecule to the next tRNA molecule. This is a little bit confusing. So if we zoom in and look at the P site's mechanism, which is what we'll do later on, this will begin to make more sense. And the final site, which is reserved for empty tRNA molecules that are no longer carrying amino acids, the E site is simply where they exit. So not surprisingly, this is what we call the exit site of a ribosome. Now, to make sense of this, we can actually take a look at one revolution of the function of a ribosome. So this all begins with tRNA that is carrying the next amino acid is ready to join the process and therefore enters the A site of the ribosome right here. Now, at this point, we need to look to the activity of the actual catalytic part of the ribosome, the ribozyme. Now, its job is to take the polypeptide chain on the tRNA molecule in the P site and break this bond here, and then transfer it to this amino acid on the A site. So here, the ribozyme's job, after it breaks off the polypeptide chain from the tRNA in the P site, it's going to transfer that to the amino acid on the tRNA in the A site, which is why we call this the peptidyl transferase site, because you're taking a polypeptide and transferring it. Now, in more detail, if we want to actually look at what the enzyme component of the ribosome does, its job is to catalyze this particular reaction, which is actually technically two different reactions here, although we can summarize it with just one here. So remember that when we are joining multiple amino acids together, we are doing some sort of dehydration synthesis reaction, which means it's going to pull a hydrogen off of the amine group and then a water from the acid group here. But when we do that, this nitrogen is going to form a bond with this carbon on the amino acid, on the bottom amino acid on the tRNA in the P site, and therefore this bond holding the polypeptide chain to this tRNA here is going to break, and the entire existing polypeptide chain is going to be transferred to the tRNA in the A site. And again, the mechanism of this is just a dehydration synthesis reaction, but it's a little bit harder to recognize. Now, at this point, that means that the tRNA molecule in the P site used to, or the tRNA in the P site that used to contain the polypeptide is now empty. So the empty tRNA in the P site is going to be pushed to exit the ribosome through the E site like this, and the tRNA that now contains the full polypeptide, which previously is in the A site, is going to be pushed into the middle of the ribosome in the P site, where it is now ready for the next tRNA to enter the A site and have the fully polypeptide transferred to it exactly as we saw. Eventually, what's going to happen is that the small subunit of the ribosome, after moving from codon to codon to codon, is eventually going to see one of three possible codons, and those three possible codons are UAA, UGA, or UAG. For those with a good memory that remember the codon video, these three codons are the three possible stop codons. So when the small subunit of a ribosome recognizes a stop codon, we have entered the process of translation termination or ending translation. Now, each of these stop codons does not actually correspond to a tRNA molecule. What enters the A site instead is actually a type of protein, and this protein is what we call a release factor. Now, the fact that it's protein and not tRNA is very important for its function because the release factor and tRNA don't exactly have the same structure. So because of that, the ribozyme, the catalytic part of the enzyme, does not actually know what to do when it sees the release factor, and therefore the polypeptide chain in the P site cannot actually be transferred to the release factor in the A site. So instead, after it breaks off the polypeptide chain, it just lets the polypeptide chain go into the cytoplasm of the cell, and then the ribosome following that inactivates and actually falls apart, thereby ending the entire process of translation. Now, in terms of how the release factor actually works, we've seen examples of chemicals that are like it. The release factor can actually fit into the active site, or at least it can fit into the A site, but because it actually causes the ribosome to stop functioning, we can technically think of the release factor the same way that we think of a competitive inhibitor. And remember that all a competitive inhibitor is a chemical that can fit into the active site of the enzyme, but does not cause the enzyme to undergo catalysis, and therefore blocks the enzyme from functioning, which technically is exactly what the release factor does. It's just this enzyme inhibitor actually serves a distinct purpose because we can't keep translating indefinitely, because each protein and each gene is only a finite length. This concludes our video series on translation. To test your knowledge of translation, you can take a look at the case study below, as well as all of the practice problems following it.